Hi guys, I'm recording this video to show to you all what we have done in this last month regarding the glucose labeling analysis. I decided to make the video because it's too much things for a meeting. Then you can watch and take note of your questions and send me, or just we can schedule a meeting to discuss other things. So basically, what we have done was to analyze the data that Alex sent to us from the standard labeled compounds, the glucose labeled in the carbon 1, the carbon 2, carbon 3, 5, and 6, to discover the composition of the fragments that we can use, and also to find a way to calculate the 13C enrichment at atom position. So we managed to do that, but I'm gonna show you. It's not a straightforward way to do. So basically, I'm going to show you some results that we obtained. For this is for the fragment 129. So glucose is non-labeled and glucose is full labeled, so it means that it has 3 carbons labeled. And when you are going to check here, we see that we have an increase in the carbon when you have the glucose labeling the carbon 4 and the carbon 6 also, and also in the carbon 3. But the main conclusion that I can take from this analysis is that this standard here is not it's contaminated, so probably has the carbons 3 and 4. You're going to see. So what we believe is that this fragment has 3 carbons and probably has the carbons 4, 5, and 6. Okay, so this is also confirmed by the GC-APCI-MS. Again, we can see an increase here in the peak using the 3 labeled carbon glucose, and we have increase here also in the 5 and the 6. So again, we believe this is it's a contamination. The standard has probably 1 or 2 carbons labeled, not the carbon 3. Regarding the fragment 133, the electron part data didn't solve what is the composition of this fragment, because this is the glucose non-labeled and this is the glucose fully labeled. We cannot see an increase. I mean, we can see some increase in the 134, but this do not overcome the 133. So, and we cannot see an increase here in the 134 in any of the positionally labeled glucose. So the conclusion is that or this either this fragment has no carbon from glucose or the carbon 4, because we are not analyzing the carbon 4. But when we analyze it by GC-APCI, we could see here one increase in the 134. So this is the non-glucose non-labeled, then this is the fully labeled. So one carbon from the glucose backbone, it's included in this fragment. So going to the positionally labeled glucose, we cannot see increase in the 1, in the 2, and in the 5, but we could see some increase in the 6 and also in the 3. So the 3, again, as I told you, it's quite problematic, because you can see here it's not clear the increasing only in the 134. So for all the fragments that we are analyzing, this happens when we are analyzing the positionally labeling the carbon 3. So, but you could see here that increase in the carbon 6, so probably this fragment here has the carbon 6 of glucose. So regarding the 157 and the 160, which is the most problematic because they overlap to each other, you can see here in the non-labeled glucose that you can see the 157, 58, 59, and when we analyze the fully labeled glucose, we could see one increase in the 161 and in the 162. So the 160 we know that has 2 carbons, so probably this fully labeled here is from the 160 fragment, and this increase here may be from the 157. So meaning that the 157 has 4 carbons and the 160 has 2 carbons, and they overlap in the 160 and in the 161. So we confirmed this by analyzing the positionally labeled glucose, by electron impact, and also by GC APCI. So pretty sure that they have these carbons. Regarding the 205, it has 2 carbons. So here is the fully labeled glucose. You have an increase in the 207, and here you have 205, 6, and 7, and here you have increase in the 207. And probably we saw also increase here in the carbon 5 and also in the carbon 6 and also in the carbon 3, but again, quite crazy because the 205 is also high. Probably this fragment here has the carbons 5 and 6. Yeah, this just to show that it was confirmed by APCI, and again the problem with the position, the labeled position in the carbon 3. Regarding the 217, we could see that he has 3 carbons, probably the carbon 4, 5, and 6. Again, we see a problem here in the carbon 3. This was confirmed by GC APCI. So every fragment that we have analyzed, I didn't show you all of them, but this is the let's say all the fragments that we could analyze in the glucose compound. And here the possible carbons that these fragments contain. For example, 103 has the carbon 6, but GC APCI showed that there is a second peak, and about 40% is the carbon 6, and the 60% is the other peak that we don't know. It's probably not probably do not contain carbon from the glucose. That's 104 overlap with the 103, so we cannot trust in them. The most prominent fragments that we believe we can use is 129, 133, 160. We could manage to use this fragment because this contains the carbon 1 and 2 is very important to the analysis. 205, 217, we could also use them. 262 and 259, they also overlap to each other. In the region of the mass spec is quite crowded, so you cannot use this fragment. We can use the 319. Actually, it's the best one. So it's the most clear fragment that contains the carbons 3, 4, 5, and 6. And these peaks with high mass to charge ratio, it's quite problematic. The intensity is not that high, and they do not follow the labeling pattern observed in the other fragments. So in the samples that we have analyzed, we cannot trust in any of these fragments. Actually, is the full molecule. So with this, we started to make the calculations how to estimate the enrichment in each atom position. So these are fragments that we could nicely see the composition of them. I mean, which carbons from the glucose we have in this fragment, but 262, it's impossible to use. I mean, one important message that we have to say, I guess, is what we can do and what we cannot do. Okay, because this fragment is really nice because you have 1, 2, and 3. For example, if you can trust in this fragment, we could use to estimate the carbon 3 based in the subtraction of the 160, but we cannot trust. So I guess this is an important information to show what we cannot do regarding glucose labeling analysis. The 3, it's also quite problematic because you have a second peak identified by GC APCI MS. And the 133, I don't know why, but the APC data is clear, but we could not see increase using the fully labeled glucose in the electron packed ionization. So I don't know why, so something that we have to think about. And why we cannot use these two fragments, because look to the place where they are, so 259, 60, 61, 62, so quite problematic. And you see here the intensity is not that high, so 259 and 262, although we know what is the composition of them, we cannot use them. We have also one overlap between 157 and 160. So 157 has 4 carbons, the carbons 3, 4, 5, and 6. And the 60 has the carbons 1 and 2 of the glucose. And you can see here the region is quite okay. So the intensity is quite high from both, from 157 and from 160. But this intensity here, of course, includes something from the M3 of the 157, and the 161 includes the WM1 from this fragment and the M4 from this fragment. So we managed to calculate, to estimate what is the relative abundance of the 160 and 161 of this fragment here. Then we discount and get the total 13C enrichment in this fragment here, because it's really important, because if you see the fragments that you can use, none of them contains the carbon 1 and 2. So this one contains, but we cannot trust, and all of the others that we can trust, there is no carbon 1 and 2. That's why we try to solve this problem overlapping between the 157 and the 160. So this is the overlap that we focus. And how we do that? So we basically took a fragment that also contains the carbons 3, 4, 5, and 6, which is the 319. And as I said, it's a very good fragment, very clear. Both electron impact and also APCI show this. And the region of the chromatogram is very nice, so it's a very good fragment. And here we are assuming that if they have the same carbons of the compound, they would have the same relative abundance of M, M0, M1, M2, M3, M4, and so on. So what we did was to correlate the intensity of the isotopologues. In this case, we took the raw data normalized by mass and Ribitol and correlated the M0, the M1, and the M2 between these two fragments, and you can see a very nice correlation. So they are positively and significantly correlated. So basing that, taking into account this nice correlation, we assume that the M3 and the M4 also has a positive correlation between both 319 and 157. So what we did next was to estimate the M3 and the M4 of the 157 based in the M3 and in the M4 of 319. So for this, we are assuming that they have a linear correlation, which fits well with this analysis here. So the calculation is not that simple. Here you are going to see some data. When you see Ni is the raw intensity normalized by Ribitol, and mass used to the destruction, and Ra is the relative isotopologue abundance. So to do the calculations, we started with a non-labeled sample, the Rhabdops control that you use in every run in Alice group. Or we transform the data 319 and 223. I mean, the whole fragment in relative abundance, so the total is 100%, and the 319, 320 has a percentage of this 100%. So and then we sum the relative abundance of 219, 220, and 321. So M0, M1, and M2. So the total has, in this sample, had 97.7%. So why we did that? Because we know that these three here nicely correlated with M0, M1, and M2 of the 157. So we assume that 157 also has the sum of the M0, M1, M2 also has 97.7%. And of course, the main aim of doing this is to estimate the percentage, the relative abundance of 300 M3 and M4 of this fragment. In the total, it's 2.3%, 2% of M3 and 0.3% of M4. So, and then with this information in hand, we started to estimate the enrichment, the relative abundance of 160 and 161 of this fragment. So we estimated that the relative abundance of 157, 158, and 9 based in this sum here. So the sum represents 9.7%, so it is included here in the calculation. And then we took the raw data normalized by mass and Ribital, multiplied by this percentage here, and divided by the sum of 157 and 159. So we are here calculating, estimating the percentage of relative abundance in the mu0, mu1, and mu2 of 157. So what we got was 76.6% for mu0, 15% of m1, and 6.2% of m2. And then we assume that, given that the correlation between these the fragments 319 and 157 is linear, we assume that the M3 M4 has the same percentage of this fragment. So, and then with this we got the relative abundance of 157 to 162. Okay. With this information, then we calculated the 13C enrichment, the relative abundance of the other fragment, the 160 and the 162. So the first thing we did was to transform the normalized data by normalized by orbital and mass, transforming this in percentage. Even knowing that this is wrong, I mean, we know that 160 has overlap with the 57 and the 61 as well. But then we discount, so we just estimated the relative abundance of the 160 and 61 of the 157. So and then we took this value here and discounted the percentage obtained previously. So what we got was almost 81%, almost 30%, 13% here, and 60.2% of the M2. So with this, and then we can calculate the 13C enrichment in the 157 and also in the 13C enrichment in the 160. But 157 we don't need it because the 319 is already a nice fragment that contains the same carbons from the glucose. So we did all of this to get the information from the carbons 1 and 2. That's why we did these calculations. So and then we applied the method. So so if we are convinced you that the calculation is right, we can use this information to estimate the enrichment in the carbons 1 and 2. And here we have two possibilities. We can discriminate the carbons, each carbon C3, C4, C5, and C6, or you can analyze by pair. I mean C1 plus C2, C3 plus C4, and C5 plus C6. So basically, in both analyses, the C1 plus C2, we cannot distinguish the both carbons. So this is a limitation. We didn't find any way to do to do this, I mean to estimate the enrichment in the carbon 1 or in the carbon 2. And then to estimate the carbons in the carbon 3, 4, 5, 6, I just took the total 13C enrichment in this fragment here, for example, and discount the total 13C enrichment of this fragment, and then we get the enrichment in the carbon 3. Because this fragment here has the carbons 3, 4, 5, 6, and this has the carbons 4, 5, 6. So discounting, and then we get the carbon 3. So we do the same for the other fragments. And if this is a kind of, I'm intended to say that this is a better way to show the results, because given that we cannot distinguish the carbon 1 and 2, I guess this seems to be more, how to say, it's more simple, and also the data, I guess, fits better in this kind of analysis. So here we took the total 13C enrichment in the fragment 160. To get the enrichment in the C3 plus C4, we just make this calculation, and we are using the fragment 205 to get the enrichment in the carbon 5 plus carbon 6. And this is also something nice. To get the total 13C enrichment in the glucose, you just have to sum the 160 with the 319. So then you get all 6 carbons labeled. So we took the data and applied this approach. This is the data from Marek, Rhabdops rosettes under 13CO2. And one thing interesting that we can see is that the carbon 1 and 2 again showed the a higher intensity, over time. So you get more enrichment in the carbon 1 and 2 compared to the carbon 3 and 4, and also in the carbon 5 and 6. So if you compare, of course, here you can, it's overestimated because it's the sum of 2 carbons, but you can see increase in the enrichment in all carbons, carbon 3, carbon 4, carbon 5, carbon 6, but there are differences between these carbons. It's not like the whole molecule has been labeled simultaneously. This is the data from Stephanie and Toshihiro. It's maize leaves under 13CO2. Again, it's the same thing. So we have a higher enrichment in the pair of carbons 1 and 2 compared to the carbons 3 and 4. And we took we have this is not published yet. It's the data from Valeria that you also saw already. And the interesting thing that we can see also that the carbons 1 and 2 are more enriched in the light. So here you have samples in the dark and samples in the light, and harvested after 10, 20, and 60 minutes in the light. So here we have the enrichment per carbon here per double C1 plus C2, C3, C4, C5, C6, and you can see there's no enrichment in the carbon 1 and 2 in the dark, but there is an increase in the 13C enrichment in the carbons 1 and 2 in the light. So probably this is the carbon mostly labeled by photosynthesis. And another thing that is interesting that is when you took so this is the Arabidopsis, this is the maize leaves, and here is the guard cells. And here's the percentage of the carbons. So we sum everything to 100%, and then we check what is the percentage of each pair of carbons in the compound. So you can see here that in both leaves, in Arabidopsis and also maize, the higher percentage is in the C1 and C2, and in guard cell is the opposite. So in the dark you have higher C3, C4, and C5, C6, and you cannot see that difference in the percentage of C1 plus C2 compared to the C3 plus C4. So maybe it's one effect of low photosynthetic rate, or given this is higher here in the dark, maybe it's the gluconeogenesis has been activated. So this is this guide cells here is under bicarbonate. So bicarbonate, when at least in animal tissues, when you label the tissue with bicarbonate, you get the enrichment in the C3 and C4. So the labeling the C3 and C4 in this case here is probably a result of the gluconeogenesis. One thing that we can show also is the total 13C enrichment in each fragment. So this is going to be, I don't know, by supplemental data or something like that in the paper, just to show again that the calculations follow the same pattern of the fragment. So this is the 205 and also the 319. So that's it. I just want to hear your thoughts about that, and let's see if you agree with all the results. Please send me your questions, and then we can start writing the paper. Okay, bye bye. See you.